Welcome to Unit C, a unit where we're going to talk about genetics and molecular biology. Here we are, getting into Mendelian genetics. 40% of our course is found within mitosis, meiosis, Mendelian genetics, and our DNA. You could be doing this great journey with Rebecca, and I bet you she's just helping you knock it out of the park. Or you might have been doing all these modules with myself. But either way, it's about you too. If you're putting in great effort, then great things are going to happen for you, for your school-based mark, for your diploma mark. But some of it's got to come back to you. Are you willing to go through these next 2 units? And it's a lot of process. You're going to be using a lot of paper. We're going to be drawing a lot of squares. And if you can do that with me, I have no doubts that you're going to see your understanding and application of these 2 units greatly increase. Like always, if you have a question, send it to info@rtdlearning.com, and I'll get on that question quickly, take your answers, send it back to you. We want you to have clarity and confidence in all that you do. Grab that data sheet, grab some blank paper, let's get going. As we begin our journey of genetics, we're going to begin with Mendel's and his different laws of inheritance, things like the laws of dominance and that. We're going to move to Punnett squares and talk about probability, lots of different types of monohybrid crosses, move into our dihybrids, and then finish with Thomas Morgan and his work with the fruit flies as we take a look at sex-linked traits. Lastly, in our genetics with a pedigree analysis and tons of practice questions along the way for you to refine your skills. Then in the second part, C3, we're going to work with molecular biology and really take a look at the structure of DNA, take a look at those scientists that contributed to the structure, talk about the central dogma of transcription and translation, mutations, and then finish off with some technology and lots of practice questions that we're going to come out of here with a great confidence we understand a large portion of this 40% of the course. As we take a look at genetics, we're going to use a lot of terms, especially genes and alleles. And I take a look here, I would call that big A by itself an allele, that little a by itself an allele, and as we set up our genetics, big A, little a, we're going to have a lot of terms for that, but that would be at a gene level now, and genes make proteins. But genetics itself is a study of heredity, passing things from one generation to the next. So we're looking at the DNA that's contained within our gametes because that's what we're going to pass on to the next generation. Like it says, genes are units of instructions on our chromosomes. So here's a gene, and they produce a specific trait. The proteins needed to make the shape of your earlobes. An allele is 2 or more alternate forms of a gene. Notice it says it's either the big A or the little A or A. So it's kind of like that 2N to N. It's a reduction. So an allele is 1 letter, a gene is 2 letters, and the 2 letters make a protein. Gregor Mendel started us off with his laws of inheritance. We're talking about an Austrian monk, the father of genetics, an incredible mathematician who used pea plants, both their phenotypes and their rapid growing, so that he could study what was happening with the different combinations, and he was looking for patterns. And his work laid the foundation for genetic study. By observing these visible characteristics, so our phenotypes, Mendel was able to establish the general principles of genetics, and these became his laws of inheritance. So let's take a look at what's going on. Law of dominance. Well, I see the law of dominance right here. Purple flowers are big P, white flowers are 2 little p's. If the law of dominance is expressed, there's my allele, it's found in a gamete, that's a big thing. Here's my little p, and notice, in our F1 generation, everything's big P, little p, and it's all purple. So there is our law of dominance. Gregor Mendel also talked about the unit characteristic. Traits are controlled by genes, and a gene must occur in a pair. And we'll talk about different names, but each of these is a pair. I kind of see homozygous dominant, heterozygous, homozygous recessive, but genes make proteins. The law of segregation. Look at that, you go from gene to allele. I see meiosis. You went from 2N, kind of, to N, gene to allele. And the law of independent assortment. Genes for different traits will sort into gametes independently from each other during meiosis in anaphase 1. So put a note there. This is another way to create genetic variation. I know it talks about linked genes, and we'll do those later on. That's an exception. But when we take a look, here's two genes. They are heterozygous, big R, big T, little t. And I want us to use the FOIL method. Big R, big T creates this one. Big R can go with little t. That creates this one. This is independent assortment. We have to do all the different possible combinations. Little r goes with big T, and little r goes with little t, and that created our 4 unique combos. So put this as something that you're going to review again. What are the laws that were established by Gregor Mendel? Let's come back to our Punnett square. In our F1, everything was purple with a genotype of big P, little p. In an F2, you cross your F1 results. So in F1, we had big P, little p, and we're going to cross it again with big P, little p, and that will form our F2. So here we can see big P, little p, big P, little p, and I'm see 1, 2, 3 purple, 1 white to give me a 3-to-1 phenotypic ratio. As we move towards the application in our genetics unit, we need to understand some of the terminology. You've heard me use on the previous slide the word phenotype. It's something that you can see, an observable trait created by genes. Is it round or is it wrinkled if we were talking about seeds? Genotype, we're looking at the letters. We need to understand homozygous can be also called a purebred or a true breeder. Alberta Learning really likes this term, purebred or true breeding, because we don't know if it's homozygous dominant or homozygous recessive. So if I said we have a true breeding radish, we have no idea if it's dominant or recessive. Heterozygous, here, can also be called a hybrid or a carrier. So make sure we understand that terminology. This makes it easy if we call it homozygous dominant, but we're not going to see that very often on our diploma. It's most likely going to be a purebred, and we need to work usually from the offspring towards the parents. Work always from the kids and figure out the parents is my pattern to make it easier, and that'll help try to figure out, was it a dominant or a recessive trait. We use our Punnett squares as a probability tool. And we emphasize probability because every time you have a different offspring, the Punnett square resets itself. So let's take a look what's going on here. I see a gene here. I see alleles here. So remember, that was segregation. Meiosis, gene to allele. And the same here, you can see the male. They even try to make these look like sperm. Gene to allele. So our dad was able to give big B, little b. Our mom was giving us big B, little b. And I see 1 to 2. So 1 big B, 2 that are heterozygous, and 1 that is homozygous recessive. So my genotypic ratio was 1 to 2 to 1. Big B, big B, little b, little b, little b. I see a 3 to 1 big I's to small i's. The Punnett square is our probability tool. Like it says, not the actual results, but allowing us to predict what type of offspring will be born. remembering that the Punnett square is a probability tool, and probability, as it notes, is the study of outcomes or occurrences. For example, when you toss a coin, it's a 50-50 chance if it's heads or tails. But there's some rules that we need to do when we're talking about probability. I like this one, the rule of independent events. A couple has 3 daughters, yet the possibility of the 4th child being a girl is 50%. And that means because the couple had 3 girls before, doesn't matter. It has no influence on that 4th one. 50% girl, 50% boy. The product rule is when you take your Punnett square, for example, find what is the probability of getting a white flower, and then multiply it by a second probability, or maybe it could be like a white cow, and then multiply it by gender, and you have to do those separately. That is our product rule. You always multiply the events separately. So example, the probability of having 3 girls in a row, 1/2 times 1/2 times 1/2 equals 1/8. The second thing on this slide is known as a test cross. And a test cross is used to determine the genotype of a parent that is unknown. Once you put that big B, the dominant trait, and let's say it stood for black, we don't know if it's homozygous dominant or if it's heterozygous. And the only way to prove that is to cross it with a test cross, a known homozygous recessive. So if we do that cross, and if we see all black in our offspring, then we could say most likely it was big B, big B. But if we see 1/2 black, and let's pretend that other color was yellow, 1/2 yellow, then without a doubt we know that our parent was not a purebred. They were a hybrid. They were a carrier. They were heterozygous. That's a test cross, using a known homozygous recessive to see if you can express any hidden recessive traits within the parent. Now, as we go through the next slides, I really want you to pause me, try it, read it, and then take a look at my solutions as you go through it. It's only about you, and we're starting simple, but it's about you trying them and really making sure that you're reading carefully and getting the correct gametes. All right, put me on pause. I'm going to start now with example 1. Here we've got the definition of autosomal, traits not affected by sex. So that's pairs 1 to 22 in humans. It also notes that you must have 2 recessive alleles to express that trait. So like little a, little a. Example 1. Short hair is dominant over long hair. 2 heterozygous rabbits are mated. What phenotypes are expected? So you should be looking at this going, okay, 2 heterozygous, big H, little h, times big H, little h. Let's put that in our Punnett square. So we've set up our Punnett square with both parents. Now, do your crosses. So we're going to have big H, big H, big H, little h, big H, little h, and little h, which is going to, and we're solving for phenotype. What does it look like? That's our question. And you should have had 3/4 of them are short, 1/4 are long, for a 3-to-1 ratio. Number 2, normal pigmentation in humans is the result of a dominant allele. White albinism is the recessive allele, so therefore 2 little a's will express white albinism. 2 heterozygous individuals plan to have a child, so we're heterozygous. So you're like, all right, that's easy. Big A, little a, times big A, little a. But the key is they want to know albino girl. 2 separate probabilities. 1 for albino, 1 for girl. So we set up our Punnett square. We get our probabilities. Look at that. 1 out of 4 will be albino. Now we're doing a second one. We're doing gender. So this is one way of showing gender. And we have a 50% chance of getting a girl. So we set that up. 1/4 times 1/2 to get 1/8. Round that up. So 0.13 if this was a numerical response on our diploma. In this type of monohybrid cross, we're going to look at autosomal dominance. We're also always remembering we're on chromosome pairs 1 to 22. So if the question says on pair 6, you must use an autosomal approach. So autosomal means it's a trait that's not affected by gender or the sex, and an individual who has one dominant trait will express this trait. So trying number 3, remember you're always pausing me before you begin this stuff. So here we've got a disease of dwarfism characteristic by very short arms, and it's due to an autosomal dominant gene. 2 heterozygous dwarfs plan to have children. So I see heterozygous times heterozygous. What is the chance that their first child will be a dwarf? And then there's the second question. What is the probability that the second child will be a normal-sized boy? Let's take a look. Setting up the first one, we got our heterozygous parents. Then we do our cross, and we can see 3/4 are going to be a dwarf, and 1/4 would be normal. That's our 2 little a's. In question number 2, we're doing a probability of both a boy and normal sized. So 1/4 times 1/2 to give us 1/8, or our 0.13 if this was a numerical response. When we look at multiple alleles, so sometimes there is what we call a dominance hierarchy. So a gene loci can have more than 2 alleles, and that statement, a range of dominance. You need to figure out what's the most dominant and what's the least dominant. So in this, taking a look at coat color. Now, when we talk about coat color, we look at, in this one, we have a D1, full color, D2, dilute, and D3, deadly. Let's see what's going on with the kids. A full colored female, so we know she's D1. And a dilute colored male, we know he's D2. But there's got to be something more here. Well, let's take a look. You keep reading and go, hey, some of the children died. It's the most recessive. So it must be D3, D3. And when you do that kind of rationale, it really sets it up with that understanding of how to use the offspring to predict the parents. Notice we've said we're not sure yet, but once we know there's one that's dead, it now switches both parents to D3. So the question is, what is the percentage of the surviving offspring expected to be dilute colored? So dilute will be our D2, D3. So we've determined the percentage of dilute from the 3/4 that survive, and we've got the 2 that are going to make it, but we only have 1 of the 3 that is actually dilute. That's this guy. So that'll be 1/3 of the survivors. Remember, we didn't count the dead one. In continuing with another type of dominance, we've got 2, complete dominance and incomplete dominance. Complete dominance is what we've been doing so far, where one dominant masks the recessive allele. Heterozygote always shows the dominant phenotype. There's an incomplete or intermediate inheritance where neither allele is dominant. So I always use the formula with my students going A plus B equals C. And this was just our language to say, hey, if you see 3 different phenotypes, you're probably looking at incomplete dominance or codominance. Now, in this example, remember, you're putting me on pause. I want you to try it. But in this example, we're taking a look if a purebred red flower is crossed with a purebred white flower, the F1 generation is all pink, FR, FW. They're asking us to do an F2 generation where we're going to cross 2 pinks, so FR, FW times FR, FW. So we set that up in the Punnett square. You do your cross. So red, pink, pink, white. So I see a 1 to 2 to 1 ratio. The second example, in codominance, both are expressed at the same time. So, and as it notes, the heterozygote is when we see both being expressed. In short horn cattle, if a pure breeding red, here's my red, bull mates with a pure breeding white cow, here's white, they all produce roan, which is both red and white hair being expressed equally. So it kind of looks like a rusty color because of the red and the white intermixed equally throughout the hair. Now it's asking us, it's looking like they're going to talk about gender. If a roan short-horn cow is crossed with a white short-horn bull, what is the probability that the offspring will be a roan cow? Remember your product rule. So let's set it up. Here I see our roan, and that's our roan cow. Here's our white bull. When we do our cross, there's roan, white, and we're searching for what is the probability that we'll have a roan cow. So roan is HRHW. I know that a cow is 1/2 or 0.5. So therefore, 1/2 for roan. So we could write it either way, 1/2 times 1/2 to get our 0.25 or 1/4. Another really good example of multiple alleles and codominance is found when we look at blood typing. On this chart over here, we have all our different blood types. I see homozygous blood type A. I see heterozygous, or another way to call that would be blood type A carrying O. So purebred type B, here's B carrying O. Here is codominance. They're both being expressed, and here is the recessive to produce blood type O. This formula used to be on our data sheets. IA is equal to IB, so that shows us the codominants, which are both greater than blood type O. Let's take a look at example 7. One parent has blood type A and the other parent has blood type B. Determine the parental genotypes if the offspring have the following blood types. Here we see it's got everything, A, B, AB, and O. Because we have O, this should be a fairly simple one to solve because O can only be produced by 2 little i's. So we set it up. We've got one parent that is blood type A now, and the second parent is blood type B, but we know the second position is going to have to produce blood type O here, so therefore both parents must be carrying the little I for blood type O, and now we've solved what question A would be. In question B, it's only showing codominance and blood type B. So we set up our parents. One is blood type A, one is blood type B, and then we know that's going to produce our codominant, both A, B being expressed. But how do we only produce blood type B? If we're going to produce only blood type B, therefore, that's going to be a homozygous blood type B, and our parent that is carrying A, you can't put another B there because that'll make it blood type AB, so they must be carrying blood type O. And when we set it up like this, we get 1/2 for AB, 1/2 for B. Now we begin our dihybrid crosses, meaning we're going to have 2 genes. And this really exemplifies the law of independent assortment. And we remember this law stated that genes on separate chromosomes assort independently during meiosis. One gene doesn't influence the inheritance of the other. So here I see a true dihybrid. They are heterozygous at both traits. Now, I encourage my students to use a FOIL method. Big R, big Y, big R, little y, and we need to find all unique possibilities. There's nothing left for that big R. So we start with our little r, big Y, little r, little y, and we're done. We have 4 unique combinations. Now, there's 2 ways you can solve it, and you've got nothing but time on this diploma. The 16 squares is a great way to go after it. Also, the product rule, multiplying probabilities from 2 monohybrid squares, and I'll explain that on the next slide. I want you to remember this trick too. if it's a true dihybrid. That famous ratio that you took in class is 9, 3, 3, 1. Well, 9 are going to be big R, don't care, big Y, don't care, and I say don't care because remember the law of dominance. It doesn't matter what that second position is. 3 are going to be big R, don't care, 2 little y's, and I'll just put a little dash so we remember they're little y's. In this one, we'll have 2 little r's, big Y, don't care, and 1 that is completely homozygous recessive. Little r, little y, little y. Remember that trick. That'll speed you up, especially when we talk about things like epistasis. When we look at our squares, and I'd like you to try that cross that we saw in the last one. So you're going to be doing big R, big Y, times big R, big Y, little y, and setting it up for your 16 squares. So if you remember how I created those gametes on the last one, emphasizing the law of segregation, we now see both our parents and our gametes are crossing here. If you're doing this using the product rule, you only put like letters on each box. So keep your R's here, your Y's here, and I'll explain how to get that 9331 on the product rule. The 16 squares is literally putting the R, Y, Y in the first box. You're going to go through that box and you're going to find that 9331 ratio. When we do it in the product rule, it's a bit different here. They both get the same answers. You're going to need to go round, 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 wrinkled. So I see 3/4 round, 1/4 wrinkled. Do that for our color, yellow, 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 green. So we have 3/4 yellow, 1/4 green. So for example, if I was asking what percentage are round and wrinkled, I'd go 3/4 for round times 3/4 for yellow, and I'd get 9 out of 16. There's my 9. What if I said I wanted to find out how many are wrinkled and yellow? Wrinkled is 1/4, yellow is 3/4, and I get 3 out of 16. If you did that for all of them, you'd get your 9331 ratio. So I've shown you 3 different ways to solve a dihybrid. This only works perfectly if both traits are heterozygous. Whenever possible, we want to see if we can avoid redundancy if things are going to be repetitious. So sometimes we don't have to do all 16 squares, but we have to look for those patterns and we need to be very careful. So for example, if a black coat is dominant to a brown coat, and a short hair is dominant to long hair, so we're using B's and H's, determine the phenotypic ratio of the following cross. Put me on pause, try it, and then let's come back and take a look at this together. I see that we're going to have some redundancies caused by the 2 big B's there and caused by the 2 little H's there. If we were to set up it in all 16 squares, we set up our gametes, remember your FOIL method, big B, big H, big B, little h, and I could do the same for this one, big B, big H, big B, little h, but there's my redundancy. So I create my 16 squares, and I can fill it in, but take a look. I actually had only 4 unique genotypes. So I can minimize that by just doing 4 out of the 16. It's the same probability. I could have also put it into the factor method, where you're going to multiply it through, and then you identify, because we're looking for black coat is dominant to brown and short hair, determine the phenotypic ratios. And here's our hair length. We can see the big H for short hair, and we've got 50% as well there. So the chances of black coat is 100%. Chances of short hair, like we said, is 50%. Chances of long hair is 50%. So based on what the question was asking, we're basically getting a 1-to-1 ratio. This is an interesting type of genetics. We're still doing dihybrids, but genes on one chromosome can affect the expression of other genes. Often we call this a masking or a hiding of the other's expression. In this example, you're going to read it. You're putting me on pause now. At least 2 pairs of genes control eye color. One pair of alleles is brown, big B, and then little b is blue, so there's our eye color. But you require pigment, so if it has a big A, it will produce the color. If it has 2 little a's, remember it's recessive, it will be albino, no color. If it's albino, it's epistasis, and it will cause the pigmented to produce albino no matter what it should have been. Determine the probability of producing a blue-eyed offspring in the following parents that are crossed. So there's my 2 little a's. Did it jump out to you that one of those parents is already albino? So we set up our gametes. We do that FOIL method. Notice the redundancy. We only need to do the 4 squares. So now, what is the probability of producing blue-eyed? I go, well, that could be blue. That could be blue. I know this is not blue because it has 2 little a's. That's albino. So I see 1 out of 4. That will give us our actual answer. Here we've got multiplying it through, and we can do the 1/2 times 1/2. But ultimately, we're going to see either method shows me 25%. So this would have been 1/2 times 1/2. That gives us this. And now we've got our 25%. All right, you've put me on pause. And now you're coming back to take a look at example number 9. Red color in wheat kernels is produced by big R, big B. Don't care about those second spots. White color is double recessive. The genotypes R with 2 little b's and 2 little r's with 1 big B produces brown. What is the probability of producing brown kerneled wheat in the F1? So in our F1, we're actually going to cross 2, in our parents, we're going to cross 2 that are pure breeders. One that's purebred red and one that's purebred white. There's my alleles from the purebred red, and there's my alleles from the purebred white. They can only produce that in our F1. Now in the F2, it wants us to take this and cross it with itself. So big R, big B, little b, times big R, big B, little b. Like it says, if you remember the 9-3-3-1 ratio. You can also set it up for your factor method. There's my 16 squares. So if you were to count the 16 squares, you would find 6 out of the 16, or reduce it to 3 out of 8, or 37.5% were brown. I prefer my method. 9, 3, 3, 1. Big R, big B. Don't care. That is red. Big R, don't care. 2 little b's. We got 3 browns. 2 little r's, 1 big B, don't care. Now we have 6 browns. Little r, little b, that's white. So there's my answer. 6 out of 16 by remembering that simple way to do a dihybrid cross quickly. Now, you've gone through, and if you haven't, pause me. I want you to tackle all these genetics questions and then come back and let's do them together. Question number 1, Mendel's principle of segregation states that alternate forms of a gene separate. So when do they separate? Segregation. That word to me means meiosis. So meiosis occurs in gamete formation. So 1 being D. Question 2. Gaucher disease is characterized by fatty substances on organs. It has an autosomal recessive. That's a big thing to remember. So it can only be shown if both are recessive. A woman who is heterozygous, so she's big G, little g, has a child with a man who does not have the disorder, so he's big G. But his mom did, so it's a hidden way of saying he's heterozygous. So let's set this up. Big G, little g times big G, little g. Put our cross in. What is the probability that the child has Gaucher disease? And I'm hoping you can look at that Punnett square, see this, and know that's 1 out of 4. So therefore, our answer is B. Question 3. The Kermode bear, also called the spirit bear, is a subspecies of black bear found in BC. Most have a black coat, but a small number have a white coat, and that's caused by the presence of 2 autosomal recessive alleles. So if you have 2 heterozygous bears mate, what is the probability that their cub is a female, so that's 1 probability, and has a white coat? That's a 2nd probability. So we set it up. Here's 2 heterozygous bears being crossed. Here's our gender, and we know we're looking for a female, so that's 1/2. So 1/4, because we were looking for the white coat, 1/4 times 1/2 for gender to give us the 0.13. Question number 4. Some chicken breeds have 3 different phenotypes. Do you remember my formula? A plus B equals C. It can be that easy on your diploma exam. A plus B plus C tells me incomplete dominance. In question 5, the coat color of Labrador retrievers is determined by 2 alleles, the B's and the E's. But the key thing here is 2 little e's prevents the expression. There's our masking, which we also know is epistasis. It wants us to do a cross between 2 that are completely heterozygous. Big B, big E, little e. So you can set this up. We know the ratio is going to be 9, 3, 3, 1. 9 have the dominant trait, 3 have the first and the second, 3 have the second but not the first, and 1 that is completely recessive. That's quite the language. So let's take a look at it in a different way. Do you see how if they all have the little e's, so now I see 4 are going to be yellow because of that 2 little e's. So 9 will be black, 3 will be brown, and 4 are yellow because of the epistasis. The Humulin gene determines coat color in dogs. Big E produces black, little e produces red, and then there's a Merrill gene that controls the expression of color. And it says Merrill is incomplete dominant. So now we're going to have to play, and here I see our incomplete dominance. We got 3 different types of colors. A dog that is homozygous dominant for Humulin. Here's my homozygous dominant for Humulin. Mates with a dark red dog. So dark red needed to be the 2 little e's. And we've got her set up for dark. Remember, it had to have the 2 little m's as well. So we set that up. We do our cross. And as you go back to your what does this produce, you're going to see that it can only produce gray and black offspring. So in continuing with the information that was given to us, we're still looking at the Humulin and the Merrill gene. We're doing something in this one called a test cross. And a test cross is where you take a known homozygous recessive. If we're going a known homozygous recessive, that's going to be 2 little e's and 2 little m's, and that's going to produce dark red. A dog breeder wants to determine the genotype of her white dog. So now we got to figure out what makes white, and they're going to use a test cross. They find out some have gray coats, that's a key thing, and some have light red coats. Let's take a look. If we know our test cross is dark red, it can only be one of these 2. Now, if it's big M, like this one, that, for the male, can only produce white. So therefore, it must be, as we take a look and do the same logic here, the gray phenotype, it must be B, where we're going to be able to produce all those different gray and light red puppies. Now for our notes, we're moving into the chromosomal theory of inheritance. This was developed over many years and can be summarized as follows. Chromosomes carry genes. Lots and lots of genes. We've seen homologous chromosomes and they segregate during meiosis, and this causes each gamete to have half the number of a somatic cell. That's 2n to n. Chromosomes assort independently during meiosis. We've seen that, especially in metaphase 1. Genes on different chromosomes are not linked. If genes are on the same chromosomes, these are linked. They are likely to be carried together. Linked genes, as it says, are inherited together unless crossing over in prophase 1 occurred. Remember, it's a removal of a fragment, and any linkage found on either the X or the Y chromosome is called sex-linked. When we take a look at sex determination, and we're doing a different type of genetics now, we're going to be working with the X chromosome. Females only have an X chromosome, and in human females, 2 X chromosomes creates a normal female. This would show the monosomy of Turner syndrome. Males have both an X and a Y chromosome, and when we look at the karyotype, here we have our 2 different genders. XX, female, XY, male. Thomas Morgan really wanted to prove Gregor Mendel's work, but he was getting different ratios. And he's like, why is this so different? And what he started to figure out was Gregor Mendel's ratios work on autosomal traits, but not when they're carried on the sex chromosome. So let's take a look what Thomas Morgan did with his fruit flies. He crossed a red female with a white-eyed male. So that should actually be a little r right there. XRY. It's correct here. Here's my red female. Here's my white male. We cross it. Red, 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 red. Thomas Morgan's like, where'd the white go? What happened here? So he's like, well, I better do an F2 cross, and I'll take the offspring from my F1. So notice he set up the male here, and he's taking these heterozygous females and placed them here. He crosses again. Red, 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 white. So he goes, what happened? He goes, there's a difference in the pattern of inheritance between males and females. And he was able to take that information and then eventually figure out, hey, I'm working with some genetics that's being carried on the X chromosome. And thus, we start a different type of genetics called X-linked crosses. All right, put me on pause. Try it. Your first X-linked cross. The first thing I see here is hemophilia is a recessive trait. So I'm just going to put X little h is for hemophilia. What do I know about this? Well, a female, to have it, must have 2 little h's because it's recessive. A male only needs 1 because nothing is going to be carried on the Y chromosome. So let's take a look what they give us. A man with hemophilia has children with a woman who carries it. Maybe they didn't tell me it was recessive, but the moment I know you can carry a disorder, it is a recessive disorder. But she has normal blood clotting time. So we set it up. Here's my male with hemophilia. Here's my carrier. What proportion of their children would you expect to have hemophilia? All right, so we go into our Punnett square. No, 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 yes. So I see, Oh, I missed one. There's my female that has it. My mistake. There's my male that has it. So there's 1/2. And what proportion of their sons? Well, if they have boys, we're only referring to this side of the Punnett square. Therefore, 1/2 of their sons is the probability of having hemophilia. As we continue to move through our genetics unit, we're now moving into pedigrees, which is going to allow us, as we see in this diagram here, to look at generations and see if traits are being expressed. And then we need to really be able to look at it going, hey, is it autosomal? Is it sex-linked? And these different things so that we know what type of approach we use when we create our Punnett squares. So on our data sheet, we've got the male, the female, mating between close relatives, but we're looking for the generations, and in particular, are they affected or not affected? When you approach a pedigree, remember, autosomal or sex-linked. You need to figure out these different patterns. If it's recessive, look for the trait skipping generations. If it's dominant, it's going to be in every generation. You can't magically make a dominant trait disappear in one and then reappear. But a carrier parent, like we see here, can actually have the dominant trait, and in the next generation, all of a sudden, if you had a big R, little r, big R, little r, it's possible to have the recessive trait show up in the next generation. With our autosomal dominant, we know they could have an unaffected, but that would only happen if they were carrying. So a dominant trait can hide or carry the recessive trait. And like it says, equally. In autosomal, look for it affecting males and females equally. If it's sex-linked, recessive, two things I look for. It's going to skip a generation, and if you see more males than females being affected, definitely look for that. That is our key for X-linked recessive. Once again, with X-linked dominant, no skipping of the generation. A female cannot carry a dominant trait, she'll express it, and no male-to-male transmission because nothing is carried on the Y chromosome. An affected father, so remember, it's a dominant disease, will always pass it on to his daughter because he's going to be giving the X with the disorder. So when you see a pedigree, ask yourself two questions. Is it dominant or recessive? Is it sex-linked or autosomal? Put those two together, you'll have great success with pedigrees. I'd like you to put me on pause and take a look at A, B, and C. And ask yourself, hey, if I was to look at this pedigree and use that language, first, recessive or dominant, autosomal or X-linked. Put me on pause, try it, then let's go through these together. As this statement says here, why do we think it's autosomal recessive? Well, the first thing I see is it's skipping a generation. So two unaffected parents can have an affected offspring. I agree. Personally, because only a male was affected, I would have started from an X-linked recessive approach. There's only going to be one answer on that diploma, but just because it only affected a male, and this is such a small sample size, I would have started from X-linked recessive. In looking at B, first thing I see, hey, it skipped a generation, and it's affecting both males and females. So, skips a generation, correct. Two unaffected parents, definitely autosomal recessive. What evidence do you see that it could be X-linked recessive? Equal male and female, but affected female's father is unaffected. Right here. So therefore, it cannot be X-linked. He's not carrying. And then in the third one, first thing I see is it's in every generation. So I'm going to go with dominant. And it could be both, and we'll explain. These are so small in pedigrees. Personally, I would have started from X-linked dominant because I see only females being affected. Remember that statement. An affected father of an X-linked dominant disorder can only produce affected daughters. So our evidence for autosomal dominant doesn't skip a generation. Affected parents have unaffected offspring. So that would tell me that they're probably carrying the little r. And because there's more females than males, that's our strong evidence that it could be X-linked dominant. Once again, put me on pause, give it a try. A question on piebald spotting. So piebald spotting is a rare disorder, and it's a dominant allele. So we know big B is going to code for piebald spotting, little b for normal. What is the probability that any offspring produced by individuals 2-5 and 2-6 would have piebald spotting? So 2-6, I look at that lady and I know without a doubt that she's two little b's because she's unaffected. So at two little b's because she doesn't have the dominant trait. And now we need to look at 2-5. So 2-5, I need to go to their parents. That's where they got the dominant trait. But notice, Dad was recessive. He could only give two little b's. So therefore, Dad must be big B, little b. We set this up in our Punnett square. So here's our 2-6. Here's our 2-5. And when we do our cross, we'd see that 50% of the offspring will be piebald. As we move into gene linkage and crossover frequencies, and remember crossing over was always in prophase 1. And as we talked about independent assortment, Mendel's law, remember how we were using the FOIL method to create our 4 separate types of gametes to produce that 9:3:3:1 ratio. Thomas Morgan, we discovered the linked genes and on the X-linked inheritance. When we're taking a look at our parental generations and moving, the big thing to note is linked genes do not sort independently. So we're going to take a look at what would cause this not sorting independently. Why would something like this happen? And it's all about placement and how close they are on the same chromosome. So when we talk about crossing over, anything new is known as a recombinant. So it was not found in the parents' combination. I like to think of it as a long stick. If you have two genes or two alleles here or two genes, if they're close to each other, if you randomly snap the stick, remember crossing over is breaking a fragment. If you randomly snap it, the ones that are closer together are likely to stay together. Ones that are further apart are more likely to be separated. And that's what this means. The closer the two genes, the less likely crossing over occurs. We have a formula here, but on your diploma, you won't need the formula. You're just going to be able to need to apply the crossover frequencies that were calculated by this formula. Remember, a recombinant is something that is different than what was found in the parents. So our recombinant, showing that we have new combinations that were not found in our original parents. Think of these percentages like centimeters. We're creating a map together. So example 11. If you've already tried these before, put me on pause. Give it a go. And for those, work with me. When we take a crossover frequency and we're calculating our gene map, the key is always begin with the longest. Put it on our map. A to B is 12 map units. If possible, always work inwards. Start there. Find the next longest one. So I see B to C. So B to C is 7 and A to C is 5. We've got her mapped, and we would say that the mapping of this one is A, C, B or B, C, A. In number 12, researchers have constructed a genetic map of the chromosomes, and we do the same thing. Let's find our longest one. 2 and 3 is 24.3. So we put that on our map. Then, if we can, we want to work inwards. So we find 3 to 4 is 16.1. We put our 4 here at 16.1, and then we find 2 to 4 is 8.2. So mathematically, that's going to add up to our 24.3. And then the question is, what is the distance of the gene between 1 and 4? So we got to do our 1 and 3 is our information. We find out that 1 is 13 units apart. So with our 13 map units there, now we can calculate what's the difference between 4 and 1. And I'm hoping that you can see that it's 16.1 from 3 to 4. It's 13.0 from 3 to 1. So therefore, our distance between 4 and 1 is going to be 3.1 map units apart. Hemophilia is an X-linked recessive disorder. Blood clotting doesn't happen normally. Parents of a boy who both have normal blood clotting, and the boy has hemophilia. So we really got to step back and go, well, dad does not have it, so dad must be normal. And mom, she's normal, so she must have one big H for normal, and she must be carrying the recessive for the actual hemophilia. To be a hemophiliac, the little boy must be X little h Y. And his parents have the normal blood clotting, meaning, just like I showed, that they must be these two here. And when you put that into your Punnett square, I'm hoping it jumps out to you that it's obvious that a sister, there is no way a girl can have it because dad has only a dominant normal gene rather than hemophilia to pass on. So in 8, our answer is D. As we look at the researchers of Memorial University in Newfoundland, they've recently discovered a mutated gene, and we continue reading it's affecting the heart and that, but this mutation is found on chromosome 3. I need it to jump out to you. You're going, wow, that is autosomal. The discovery of the gene on chromosome 3, if it's autosomal, and it says it must be dominant here, it must occur with equal frequency in males and females. That's what autosomal means. In question number 10, Tay-Sachs disease is a hereditary disease that kills 1 in 360,000. Children who are homozygous die at an early age. Genetic screening can be done if you're a carrier. Well, if you're a carrier, then we know it must be a recessive disorder. You cannot carry a dominant trait. So therefore, what type is this? Because it's not linked to chromosome 23, this will be known as autosomal recessive. Now, as you did this chromosome mapping one, I'm really hoping that you took, just like we did earlier, drew it out, started off, hey, I'm going to start with my 2 longest ones, and you set it up so that you had your P and W and your RL, noted that it was 95 map units, and continued that process, and this should have been a very straightforward question to get the order of PNW, LA, GW, and RL. This is an excellent pedigree to take a look at. What jumps out at you? Firstly, I look at this and I go, without a doubt, I'm looking at some sort of dominant condition. It doesn't seem to skip any generations. The second thing that I note is in a very quick tally, I go, boy, you know what? Looks like it's affecting males and females pretty much equally. So let's see what we got. It's a tooth condition. A student is trying to identify the correct mode of inheritance, and the student thinks it's autosomal recessive. All right, autosomal recessive. Once I see autosomal recessive, I'm drawn to here. And why I'm drawn to here is, imagine it. Little d for 2-3, little d for 2-4. Well, how then did you produce 3-7? Noting, all of a sudden, that trait appeared. But if we switch that so that the parents were now a dominant condition, then we can produce all the offspring, 3-5, 3-6, 3-7, and 3-8 with this combination. So number 12, our best answer is D because of the rationale that I showed you here. Question 13, the Kermode bear, the spirit bear. We continue reading and we note its autosomal recessive is going to cause the white. So a female bear with a black coat gives birth to 2 twin cubs. 1 black, 1 white. Which of the following rows identifies the genotype of the female and the genotype and phenotype of the male? Well, the first thing I'm going to do is get rid of C and D. Both of those are showing me X-linked inheritance. So we know the mom is black and has twins. So that has to be created by both parents. When we set it up to produce that twin, the only combination that we have that's going to work, because note here, the female could never pass on the little b, so 13 must be B. Alright, in rare cases for number 14, single gene mutations can cause obesity. The mode of inheritance can be autosomal recessive, autosomal dominant, X-linked recessive, or X-linked dominant. So it can be all the genetics that we've covered so far. And they're asking us, and this is a really good one for you to review again, do you know without a doubt your 4 types of inheritance? So autosomal recessive. So I know it must be homozygous for this type of trait. So right now I go to my list. To be affected with this form of hereditary obesity, a person must be homozygous. That is true. But I haven't gone through all of them. Let's see if that sticks. Autosomal dominant. 2 unaffected parents cannot produce an affected child. 2 unaffected, because they're recessive, cannot produce an affected. Yeah, that's true. Because if they were both 2 little d's for disorder, they could never produce the dominant disorder. So therefore, 2 works really well there. X-linked recessive. We're looking for something where males are more affected. Number 4. A male requires only 1. So 3, 2, 4. And let's see. X-linked dominant. All daughters of a man who has this disease are affected. Perfect. 3, 2, 4, 1. The order in which the 4 genes listed in the legend above are located on chromosome 6. So you're going to draw your map starting with diabetes, so 1 and 2. You should have 1, 4, 3, 2. If you're struggling with this, just remember, info@rtdlearning.com. Boy, fire off an email. It'll probably be me that'll respond pretty quickly, and I can draw it out for you and send a picture of what I've done to make sure you've got it. But you should always start from the longest, and that should have been an easy one. Now, what is the approximate crossover frequency between the two? So with your map in front of you, you better be seeing that the distance between them, so we got our 21 and our 19.5, and we're looking for the distance between 1 and 4, and that will be 1.5 map units. In number 17, another genetic disorder in which the skin and joints are more elastic than normal. And the pattern it has is shown that we need to find an autosomal dominant mutation. So what should jump out to you is in that dominant, it will never skip a generation. So I look at B and I look at C, those definitely are not on my radar as correct. So which of the following pedigrees shows the most common form of inheritance? In C and B, like I said, it skips a generation. This cannot happen where 2 unaffected parents have an affected child. Therefore, the only one showing autosomal dominant is A. In question 18, we're looking at a pedigree of faulty tooth enamel. It's an X-linked, it's a dominant X-linked disorder. So XE will cause the faulty tooth enamel where it's going to create very thin enamel. The genotypes of 2,6 and 2,7. Oh, and 3,7. Well, I look at that male first. We know that the male does not have this disorder. So the male must be X little e y. If we set it up like that, our answer kind of just pops out at us. But let's prove it again. Let's go take a look at 2,6. This female produces unaffected offspring. So we know that she must be carrying, especially for that daughter, she must be carrying the X little e. So 18 being B. Number 19, a woman who is heterozygous. So you're going to set up your Punnett square, X big E, X little e, times a man with normal tooth enamel, X little e y. And when you put that into your Punnett square, what is the probability that their first child will be a boy with normal tooth enamel? So we take a look, we've set up our Punnett square, and our boy with normal tooth enamel, 1 out of 4, or 0.25. When we take a look at question 20, which of the following statements best describes the inheritance of an X-linked dominant disorder? So we had a few things. When I remember and talk about the male, I'm like, well, if it was a dominant disorder, I'm going to look at all of his daughters and they will have this disease. So you'd see them colored in. But as I look through the different things, a man who has a disorder has a 50% chance. No way. It's going to happen. He's going to pass it on. A woman who has heterozygous for this disorder has a 100% chance of passing it on to her son. No, she's heterozygous. She has a 50%. A woman who's heterozygous has a 100% of passing it on to her daughter. No, she's heterozygous. Let's check A. A man who has the disorder has a 0% chance of passing it to his son. That is correct. He's giving the Y, so therefore A is our answer. We're now moving into the last part of our Unit C. Moving into the molecular biology where we're going to talk about DNA, then we're going to talk about replication, transcription, translation, and mutations. So let's really get a strong basis of what DNA is and then talk about the scientists that helped discover it. In looking at our notes here, genes. These are segments of DNA with instructions on them. The whole point of a gene is to have the instructions to build proteins from amino acids. Our DNA, which is going to code for the instructions, is a polymer composed of 4 different nucleotides: adenine, guanine, cytosine, and thymine. And we're not going to put uracil in our DNA. That is when we talk about RNA later. There's 2 families, the purines and the pyridamines, and all DNA is found in what we call a nucleotide. So this will be its base format. And a nucleotide has a phosphate and a sugar, deoxyribonucleic. So here's our sugar, deoxyribose. And I also want us to call this part, the phosphate and the sugar, this is going to be known as our backbone. And then the only variable part is here, the nitrogenous base. So that could be A or T or C or G. When we were trying to figure out how DNA was bonded together, we owe this to Chargaff. He discovered that within a segment of DNA, the number of purines equals the number of pyridamines. This became the base pairing rule. And he noted that A bonds to T, G bonds to C. So therefore, we always go with this as our base pairing rule. So example 13. Put me on pause, read it, and let's do it together. If 17.5% of the bases in a sample of DNA are thymine, what are the percentages of the following? So we know 17.5 equals thymine. Therefore, you're going to have adenine bonded to it, and it must also be 17.5. That gives us a total, 17.5 plus 17.5, a total of 35. So then I know that we have 65% of the solution left over. Our cytosine and our guanine will each get half of that. So that'll be 32.5 and 32.5. Yeah, some of our questions on the diploma are this simple. When we look at the scientists, we're going to start off with Rosalind Franklin. And this woman should have had much more credit for the structure and shape of DNA, but unfortunately she was dying from cancer. But she shared her research with Watson and Crick. So whenever you see the term Rosalind Franklin, I want you to look for the term X-ray diffraction. She shared this image to help Watson and Crick, who actually never did any experiments themselves, but they are the ones that we credit for creating the first 3-dimensional model that showed DNA represents a twisted ladder. So Watson and Crick are credited for discovering the shape of DNA. Now, as it talks about, we've got complementary and anti-parallel strands. Complementary means this. T bonds to A, G bonds to C. That's our complement. One strand is going to be, there's my original, there's my complement. So the T, the original, the complement is the A. Anti-parallel, we talk about directions. So one strand of DNA is going to be read in this direction, and the other strand of DNA is going to be read in the opposite direction. And your teacher might have talked about 5' to 3', and then my other anti-parallel would be 5' on this side to 3', always reading in that direction. All right, here's a little bit of practice for you. Here's our original strand of DNA, and we know it's going to be read in this direction, 5' to 3'. I want you to pause me and create the complementary strand. Remembering, it's going to be 5' to 3' in terms of how it's read. And can you remember, A bonds to T, C bonds to G, and you should get this as what your pattern was. A to T, T to A, G to C, C to G, A to T, and so on. You've now created the complementary strand of DNA. What types of DNA are there? We're talking and really focusing on the DNA within the nucleus, so our nuclear DNA. But it was discovered also that mitochondria have their own genetic material, or mitochondrial DNA. And the mitochondria, interestingly enough, divide and replicate independently of the cell. They do it on their own. Mitochondrial DNA can be used to trace inheritance through the maternal lineage. So I often tell my students, I know that stands for mitochondrial DNA. Why not call it maternal? Because we know that the unequal dividing of the cytoplasm in oogenesis and the organelles, it is the mom that is going to provide the organelles, or the mom is going to provide the mitochondria to her offspring. So that's a big one when we're tracing the lineage. Like it says, the mitochondria from the sperm are not contributed, not. Hence, everything comes from the mother, maternal mitochondria. As we begin question number 21, I know you've got the pattern, you pause me, you try them all, and then you listen to the key. Here we're looking at how did Watson and Crick develop the model and structure of DNA. Like it says, they performed no experiments themselves. They used a trial and error approach. They worked out the structure as one would solve a puzzle, given all the other experiments that they were reading from people around the world, and remembering Rosalind Franklin and her X-ray diffraction were critical. The following statements are about DNA made prior to 1953. We are looking for what statements support how they learned what the structure of DNA was. So let's go through our statements. Number 1, chromosomes and genes are composed of DNA. Nothing to do with structure. Number 2, X-ray diffraction shows it's a spiral helix. Definitely. Number 3, all diploid cells contain the same amount of DNA. Nothing to do with structure. It's composed of a phosphate, 4 nitrogen bases, and a sugar. For sure, structure. It's found mainly in the nucleus. Nothing on structure. It suggests that thymine pairs with adenine and guanine with cytosine. 2, 4, 6. Which of the following rows describes a DNA molecule? Well, first thing I'm going to do is cross off amino acids. We're not dealing with DNA there. So a phosphate, a sugar, and a base. So it looks like both C and D are correct there. Sugars and bases as the backbone. Well, I know it's not bases, it's sugars and phosphates, so 22 is D. And 23, if guanine and cytosine make up 56% of the DNA molecule, what's left for adenine? So if we use 56, therefore we have 44% left, so 22 will go to thymine, and 22 will go to adenine. Question 24. Both males and females can be affected by mitochondrial mutations, but only females can transmit genetic mutations to their offspring. For this type of inheritance, so only females can transmit it. Which of the following rows identifies the contribution to the zygote made by the sperm? Well, the sperm is nuclear contents only, and the egg is nuclear and cytoplasmic. Therefore, 24 is A. When we talk about the central dogma of DNA, we are looking at two separate processes. So I like to draw a line here. Replication is going to occur in interphase, specifically in the S phase. Expression is going to take place not within, it's got nothing to do with replication. Replication is going to occur within the nucleus. Transcription and translation, transcription is going to be the nucleus, and translation is going to be in the cytoplasm and the ribosomes. So let's make sure we keep both of these separate. When we call this an expression, we are making proteins. We are expressing the gene. So replication, nothing to do with transcription and translation. When we take a look at replication, so the synthesis, like we said, is happening in the S phase. I need us to understand three enzymes. Helicase. Helicase is going to break apart. If we look in between our ladder rungs, that's a hydrogen bond. Helicase is going to snip or break the hydrogen bonds between the bases. This allows them to separate. Notice separation here. And it's going to create what looks to be like a replication fork. The parent strand, so in this case the blue strand, remains intact, and then this gold strand now comes in and serves as a template. This will determine the sequence of the nucleotides, and we also call it semi-conserved because notice it still has 1/2 blue, the original, and 1/2 new, the template. As this is all going on, we're going to have another enzyme called a polymerase. And a polymerase's job is to proofread. As the template is forming and attaching to the original DNA, polymerase, the enzyme, always ensures A bonds to T and C bonds to G. The last enzyme, notice the hydrogen bonds have been reestablished. That is our glue, our ligase. So the ligase will reattach the hydrogen bonds. So in our bottom diagram, I see here, from number 1 to number 2, I see helicase has separated. Now our blue, so our templates are going to come up. So if I had the letter A here, then a blue T is going to attach to it. And it's going to create our semi-conserved rungs. So we could have had the enzyme polymerase being a proofreader for both of our strands. And I note that they're reattached here, so that would have been by our ligase. So our goal, as we talk about gene expression, which ultimately is protein synthesis, is that we're going to go slow. We're going to really make sure that we understand both the process of transcription and translation. We can see here DNA, remember everything here is occurring within the nucleus. Transcription is going to involve DNA and mRNA. Translation, we're going to see, and it says RNA, we're going to need to learn the roles of mRNA, transfer RNA, and amino acids. Noting that this is going to take place in the cytoplasm and the ribosome. So two distinct processes. When we look at the whole process, and that's what this is, transcription and translation, when you look at this diagram, I'm just going to be particular and add an M there for messenger RNA. But in this part right here, that's our transcription. The messenger RNA is reading a gene. That's that location where it's opened up on the DNA. Once that gene is being read by a single strand of mRNA, it's going to move through the cytoplasm. It's going to come here towards the ribosome. And then with the work of what we said, the mRNA, the transfer RNA, and the amino acids, we're going to start building the protein, that peptide chain. So we're going to do it in steps to make sure that we truly have a good understanding. But that's an excellent diagram that I encourage you to come back to. It has both transcription and translation on that picture. Quick activity at the bottom. How is RNA different than DNA? Put me on pause. And then, when you're ready, let's do it together. Whereas DNA is double-stranded, RNA is single-stranded. DNA has deoxyribose, RNA has a sugar called ribose. DNA has A, G, C, T, and in RNA, there is no T. It is being replaced by U for uracil. As we noted before, DNA can be found both in the nucleus and the mitochondria. Remember mtDNA. Is it mitochondrial DNA or is it maternal DNA? While RNA is motile, meaning it can be found throughout the cell. There's 3 types: messenger RNA, transfer RNA, and you might cover this one in university depending on what you take, but that the ribosomal RNA will not be mentioned or tested on this diploma. One new thing that I'd like to mention is triplet. It always works in groups of 3. Notice this is messenger RNA. So if I took a group of 3, like I'm circling right now, at a messenger RNA level, we are going to call a group of 3 a codon. The same at transfer RNA. A group of 3 at transfer RNA level is called an anticodon. Always a triplet code. When we look at transcription in more detail, so here's my messenger RNA. We know this is taking place within the nucleus. The first thing, DNA unwinds. Second thing, I don't care what type of polymerase it is, it's a proofreader. So in this case, it's this green enzyme that is going to add nucleotides to the base pairing rules, ensuring A to U, C to G. And when the enzyme comes to a termination sequence, it stops, and then our mRNA will leave the nucleus. So from here to here, that is a gene. Genes code for proteins, and remember, transcription takes place in the nucleus. So here is a short activity that I want you to try, taking it from a DNA level to an mRNA. Before you try that one, if I had C, G, T, and I know it's now at a DNA level because I've got a T, if they don't give us a second strand, we're assumed the strand is given to us is the one that we're reading from, our coding strand. So this will be our coding strand. And we go from DNA to messenger RNA. That's our next level. So ask yourself, C bonds to G, G bonds to C, T bonds to A. Try that activity, and then I'll put up the key for it quickly. When we convert TAC, we should see AUG. If you got this right, it would have been UGC, AAU, AGG, and UCC. I want you to go to the title of your data sheet and notice how it says messenger RNA and its corresponding amino acids. What that means is, as we go through this activity, and I'll emphasize it again, if your mRNA level was AUU, then your amino acid level, it should be the exact same letters. If you've done it right, your amino acid letters will be AUU. That's key to be looking for any errors. If you're going through this, make sure your mRNA and your corresponding amino acid letters are identical. The conversion of mRNA into a polypeptide chain is called translation. We know that. It's going to take place on both the ribosomes and the cytoplasm. So let's look at our diagram down here. Here I see messenger RNA. Messenger RNA, you're going to say, I remember that a group of 3 is called a codon. Notice, G is attracted to C, A to U, U to A. I'm not going to worry about that. That's just a carrier tag on our transfer RNA. And you go, hey, I remember what was said on the last slide. Messenger RNA, CUA, is the same as it will be for the amino acid. So without a doubt, leucine has the letters CUA. You can prove it to yourself by going to that chart. Every time the anticodon attaches to the codon, it releases the amino acid, and we see our peptide chain grow. And there's a little peptide bond between each of those amino acids. Like I said, the anticodon and the codon, when they bind, it will release the amino acids. There's 2 sites within the ribosome that this is occurring, and all of this will continue until you reach one of these 3 different stop codons. It's like a period in a sentence. Once you reach a stop codon, do not continue translating. All right, definitely. Let's read the instructions together. Then I really want you to pause me and take some time and make sure you got your data sheet right beside you so that you can jump into those 64 squares and figure out what type of amino acids we have. Using that table on your data sheet, translate the following mRNA sequence, remembering it's a codons, into an amino acid sequence. All right, good luck. In knowing that mRNA and amino acids have the same letters, you should have been going right to your chart, looking up AUG and seeing that it's methionine, CCC and seeing that it's proline, UCU, serine, and so on. You should have been able to get all of these amino acids correct. Here's my stop, UAG. If there had been AAU or something afterwards, you do not translate it. Once you have a hard stop, you stop. Let's take a look at the next table. This table shows the gene expression of a DNA sequence into its polypeptide. Fill each box. So let's start at the mRNA level. If you have TAC, you should be able to go, without a doubt, AUG. If this is GUG, you should be able to go CAC. And we continue, GTG, CAC. This time we're reading from here. G to C, A to U, C to G. And we go through, and I'm hoping that this exercise was quite easy for you. The transfer RNA codons, A to U, U to A, G to C. And you continue that pattern, and you're going to get really good at this pattern, and we're going to use it a lot on the upcoming practice questions. Then that last amino acid sequence, translate. Remember, you're reading from the mRNA level. So you go up to AUG, you find out it's methionine. Then we're reading GUG, valine. And I'll just tap the rest of them in, make sure that yours match mine. And if you found this pretty easy, notice the stop codon and another stop codon. We wouldn't even actually have done this one because we stopped here. So please remember that. Once you hit a stop, you're fully done. Now, a quick review of what we call the central dogma. We're looking at the protein synthesis part of it, so the transcription and translation. Transcription, we know, is DNA to mRNA, this taking place within the nucleus. Translation is going to involve mRNA, amino acids, polypeptides. Let's throw another one in there. Transfer RNA as well. Where is this going to take place? In the cytoplasm and the ribosomes. So let's take a look at what's happening here. We get it. There's our DNA template. mRNA, group of 3, codon. Ultimately, that codon is going to produce the amino acids, but for our own reminder, let's make sure we remember the tRNA and the anticodon as we go through and summarize what's happening in translation. When we talk about a change or a mutation, it's only significant if it happens at a DNA level. If you have a mutation at an RNA level, that affects one protein. If you have a mutation at a DNA level, it affects every protein that's going to be made. It's kind of like if you think about photocopying. If you go to the photocopier and you photocopy 10 sheets and you spill coffee on one, well, 9 perfect proteins, 1 ruined one. If you spill coffee on the original, then every protein or every copy that you make afterwards, it is now an abnormal protein. A mutation can be spontaneous, can happen during replication, or it can be induced by mutagens or mutagenic agents. So we have lots of mutagenic agents in our environment. So a mutagen can be physical or chemical. Physical could be an X-ray, UV, cosmic rays, viruses, where a chemical agent could be something like asbestos, dyes, cigarette smoke, paint fumes. And the key thing is mutations are actually important too, because they allow us to change and add genetic variation. They can be positive or they can be negative. And sometimes they're just silent. So there's 3 types of mutations. There's 2 types of mutations that we're going to talk about. Germline mutations and somatic mutations. And I'm just going to put a star by germline as being the most significant because they can be passed on to the next generations. So like it says on the left, mutations can occur in somatic cells. So that's every cell of your body, but not your sperm or your ovaries. Somatic mutations are not passed on to the next generation. So that mutation, let's say it's cancer. It's a somatic mutation, but it's not present within the gametes. But if a mutation occurs within the gametes, then it's passed on to future generations. And remember, that process of mitosis is perfect. It's going to copy that mutation into every cell after the zygote. And lastly, to be inherited, the change must occur either in the nucleus or in the mitochondrial DNA. And if we're talking about mitochondrial DNA, that's only significant if the mutation happened in the female. Examples of different types of mutations. So, a base pair substitution. Let's look at our DNA level. We've really made this diagram easy for you. Cat, 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 and so on, and all it does is produce histidines. If we replace one A with a C, we still have histidine, 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 proline. And you might be like, wow, that's not too significant. That's only one amino acid that was affected. And you could be right, but sickle cell anemia is only 2 amino acids different than from what you and I have. So we can't judge the severity until we truly know what protein is now produced. An insertion mutation. Notice we're adding the A here. Everything after that addition is going to be thrown off. Threonine, serine, serine, serine. But really emphasize, because we've changed the order of all those codons, the original stop codon now is shifted. This might be a large, useless protein because the original stop location is no longer recognized. So that's an insertion. Everything afterwards is affected. Likewise, if we do a deletion. If we remove an A, just like that insertion, in the deletion, everything afterwards is affected, and no longer do we have that stop codon. It could happen earlier, but most likely it's later, and we have a large, pretty much useless mutation, or a useless protein. Silent mutations means there's no change in the amino acid sequence. That's a great thing. Missense, one amino acid is changed to a new amino acid. Well, that can have drastic effects. Frame shift, so this is both our insertion and our deletion. Many amino acids, like we demonstrated, are affected. Nonsense, when it changes to a stop codon. And that switch to a stop codon, think of it, you had a normal sentence, but you put a stop or a period in the middle. Now everything afterwards is affected. With a silent mutation, it's neat to see that if you had CCC and CCG, if you were looking at your chart, they code for, and I don't have mine in front of me, but they would code for probably the same amino acid. That third pair, that third spot, often has no effect when you do your substitution. As we move through the last parts of our Unit C, we enter to gene therapy and biotechnology. Biotechnology is referring to the use of organisms or biological products for commercial and/or industrial uses. So this could be something like the mass production of insulin, where we have now taken and put the insulin gene into a bacteria and they are mass producing human insulin. How is that done? It's done via a process called genetic engineering, in which we alter the organism's genome by selectively removing or adding or modifying the DNA. We can also create what we call recombinant DNA. So where you have two or more species and you mix the DNA. So a lot of what we have here in Alberta, a lot of the wheat and our plants are frost resistant, or they might be drought resistant. And we've taken genes from other plants or other organisms and inserted it into our wheat. So a GMO, a genetically modified organism. How do we do this genetic engineering? We have two tools. Restriction enzymes are going to open and cut open the DNA. The ligase reattaches those hydrogen bonds. It's our glue. So we're going to look at a technique on the next slide where I'm going to emphasize cut, cut, glue. Here we've got gene therapy, where we're going to insert DNA. Let's pretend this is a bacteria. And this foreign DNA, it's foreign to the bacteria. We'll call it human DNA. And we are going to isolate the gene for insulin. Well, the first thing that we need to do is take this vector. So the DNA is found in a circular plasmid. We're going to take the vector and we're going to cut it open. You can see it cut open here. That was done by a restriction enzyme. We then go to our foreign DNA. We do the same thing. We take our restriction enzyme and it's going to cut at specific locations. It might only cut after every combination of CCTA. Once it sees CCTA, it cuts. So we're going to get our different fragments. This will be the gene for insulin. We've done two cuts, a second restriction enzyme. We then place it within that plasmid vector that's opened up now because that first cut, and we're going to use the ligase, and we chose the bacteria because of its ability for asexual reproduction. It is going to make identical copies of itself, many identical copies of itself, and that's going to allow it to mass produce the human insulin because it has the gene for it. So within those vats, we can now help the diabetics by giving them actual human insulin versus previously we used to use insulin harvested from pigs. The last technique that we're going to talk about involves a jello electrophoresis, and sometimes, and I'm sure you've heard of us doing what we call a DNA fingerprint. And we know that your DNA, my DNA, and that of other species, there are parts of our DNA that are completely unique to us. And we can use them for crime scene investigation, for poaching of animals, or just creating maps trying to understand the sequences that are occurring. The jello electrophoresis is a technique where we first use our restriction enzymes. We're going to cut the DNA into fragments or segments. They're going to have different lengths. We then put them into an agarose gel that has a well where we insert the samples of the DNA into the well. So with our wells, we now run an electric current through it. Because DNA is negatively charged, the shortest strands, so we put them all here originally. Here's my well. The shortest strands are going to be attracted and easiest to move. They'll move the furthest. The heavier strands will remain closest to the original well. This will cause the separation, and we can see the distinct bands that are happening. And lastly, when combined with a staining or X-ray film, we can now create the DNA fingerprint. And we could use it for, like I said, if we wanted to see if it's a crime suspect, the DNA that was left at the scene versus one of the suspects, do a DNA analysis through the gel electrophoresis. And if it's an identical match, which it would be if you have the right person, because that DNA is unique to them. So we run all of that technique, and we can see with that gel electrophoresis, we would have now created the wells and everything moving across. You can use a gel electrophoresis to determine what's happening at a crime scene. So at the crime scene, we collected a bunch of DNA. We can see that the victim has provided this DNA. If we looked at suspect 1, it's definitely not them. None of the DNA matches up. But suspect 2 very clearly is providing the DNA samples that were not provided by the victim. So in this activity, we'd know without a doubt suspect 2 is the guilty one. As we always note, make sure that you put me on pause, giving yourself a lot of time to go through all the questions, and then when you're ready, play it, and I'll walk you through and make sure that you and I are seeing the same thing with each of these questions. For question 25, researchers are developing a technology to test for the presence of different flu viruses. One step involves using RNA as a template to produce DNA. The DNA is then cut into fragments. Reading the thing, what technology is used to cut DNA into fragments? I know it's not a ligase because that's my glue. It's a restriction enzyme. And what describes a property of DNA? Well, it can't be uracil. That's found only in RNA. So 25 is C. 26. Other researchers are testing for the presence of the different flu viruses using blood samples. With the flu is applied to a chip, viral fragments in the patient's blood will stick to matching fragments on the chip, thus identifying the specific flu virus that has infected the patient. Will stick. Well, remember, A bonds to T. So I'm looking for something that's going to be talking at a DNA level, and we're talking about the complement. How does it stick? It's when it would not stick if it comes into contact with an identical base. It would actually repel each other. But a complementary base, they certainly will be attracted to each other. So when a base in a viral fragment comes in contact with a complement. Question 27. In DNA replication, there's 2 strands, and a new strand forms along each of the old ones. Each new DNA molecule has 1 old strand and 1 new strand. What is the name of the old strand? It's certainly not haploid, so it's a template. And where does DNA replication occur? Remembering it always occurs in the nucleus. 28. Male mussels of the genus inherit their mitochondrial DNA from both mom and dad. That's different than humans. The inheritance of mitochondrial DNA in humans differs in that the mitelless mussels, because human mitochondrial DNA is, and without a doubt, it should jump to you, only goes from mom to offspring. Question 29. Processes in protein synthesis and place them into order. So we really need to know what's happening and where it's happening when we're talking about transcription and translation. So the first one that jumps out to me is the mRNA is attaching to the DNA. So I'm going to guess a 3 is going to start us off here. Then we're still dealing with transcription. The mRNA leaves the nucleus. Then what comes next? Are we making a polypeptide as we do in number 2? Or the transfer RNA is going to carry it? Well, the transfer RNA is going to carry it first, so that'll be a 4. So a 4 and then a 2. So 3, 1, 4, 2. In question 30, viruses are simple pathogens that can reproduce only within a host cell. There's about 200 and they cause a wide variety of diseases. A key statement is they contain RNA. So you really need to remember that RNA is single-stranded and it can move throughout the cell. And it's active only when it's in a host cell. So let's take a look at that. It cannot be a double helix. It's RNA. It can't have thymine. Let's take a look at B. The genetic material is a single helix. I like that. And the genetic material is a double helix. That's exactly how it's working if we were to talk about a human host. Question 31. We look at what's happening. It's protein synthesis in an animal cell. I'm hoping that you look at process 1 and go transcription. Process 2, translation. Which of the following rows identifies the structures and processes of protein synthesis as shown in the diagram to the left? Structure 1 is not a protein. It's a gene. And process 1 is transcription. So already, by step 2, we've got our answer. Question 32. A section of a gene, and they want us to get it to the amino acid level. So we must work and make sure we get to the mRNA level. Well, this without a doubt is DNA. So we go C to G, T to A, T to A. And when you set that up, you're going to get our first, you go to your chart, is glutamate, so 6. And then T to A, so A, C, U. Go to your chart, and you're going to get 309. Continue that pattern, and you'll have 6, 5, 1, 3. Question 33. What is the strand of DNA that would code for this portion of insulin? So they've given it to us at the amino acid level. So we're going to have to do a couple of things here. First, we need to go to our data chart and find phenylalanine. When you go to phenylalanine, you're going to see that the phenylalanine comes up as either UUU or UUC. Therefore, when I convert it to DNA, U to A, U to A, U to A, and then AAG as my second possibility. So immediately, we can remove C and D. We continue, and we go to asparagine. The valine was no different, because notice, CAA, CAA. I jump to this one because I see TTA and TTC. So AAU codes for TTA, that works. Or AAC codes for TTG, we don't have that choice. So therefore, our answer in 33 is A. Question 34. The amino acid sequence in a protein determines. So what does amino acid sequence do? You're building a protein. So it's going to be the structure and the function of the protein. Question 35. The anticodon of the transfer RNA molecule that transports the first amino acid. So here is the DNA level. The first amino acid by this portion of DNA. We're going to set it up. We go to our mRNA level. C to G, A to U, T to A. Remember, that's an mRNA. Now we go to our transfer RNA level. C, A, U. We set it up like that. 35 is C. 36. Variation of traits in organisms that reproduce asexually. Remember, asexual should have no variance unless a mutation happens. So 36, D. 37. Some forms of deafness are caused by mutations in the connexin gene 26. One mutation results from the deletion of 9 base pairs. Remember, our code is triplet. So you should have no problem saying if we have 9 base pairs, therefore we've lost 3 fewer amino acids. Another mutation in this gene involves the deletion of 2 bases and their replacement by 2 new bases. The deletion is shown to the right. The 2 deleted bases are replaced by adenines. So we went from A, T, C. Now we are A, A, A. The transcription of this mutated gene above results in the replacement of. So what did ATC code for? So you're going to go A, I'll just put it here, ATC, AU, AG. Go to your chart. You see that it's a stop codon initially. And now if we go AAA and you do U, 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 it's a phenylalanine. So 38 is C. 39. Metabolic syndrome is a genetic disorder with symptoms of hypertension, cholesterol, lower than normal blood magnesium, and it's caused by a mutation in mitochondrial DNA in which a thiamine is replaced by a cytosine. So we read the statement. Which of the following rows identifies the mutated DNA if we started with TCG? Well, remember, thymine is replaced with a cytosine. So we're going to go now to C, C, G. So immediately, it cannot be A or B. So mRNA is going to be G, G, C. So perfect, it could be either C or D. And then our transfer RNA is going to be C, C, G. Therefore, it has to be C. Question 40. A bacterium has been found that synthesizes a type of plastic. Researchers can remove genes from this bacterium. They cut. Immediately I think of restriction enzymes. And then insert. So remember, cut, glue. So enzymes used on the bacterial DNA. So we're moving it from a bacteria to a plant. Okay, so we are going to cut the bacteria. So I look at B and I look at D and I go, both of those are right now a possibility. Now, as I look at B, I know this is not possible. Remember, cut, glue. You have to cut open the plant DNA before you can insert it. So 40 being D. 41. To investigate a sexual assault, a forensic scientist can collect a semen sample and analyze its DNA content at the lab. This DNA fingerprint, and they're doing a comparison of 3 suspects here. As a result, 2 suspects were released and a 3rd was charged. The use of this new technology is valuable for conviction, with the exception of identical twins. Why? Because every individual is unique. Where? Not in our types of nucleotides, not in our types of proteins. Types of nucleotides would mean you got something other than A, T, C, G. We all produce similar proteins. It's not our protein sequences that we're analyzing, it's our nucleotide sequence. What order are our bases in? And with that, we have just finished 40% of the course. We began this journey in Unit C with mitosis and meiosis. We talked about alternation of generations. Then we moved into Mendelian genetics. Then we talked about X-linked inheritance. We did some gene mapping. Then we got into replication and transcription and translation, mutations. 40% of your mark comes from Unit C. I can't emphasize how important it is to have a mastery of this unit. Please rewind, go to those questions that you're like, boy, I'm not quite sure yet. Listen to that content again, whether it's Rebecca or myself. And if you need, please, info@rtdlearning.com. We have covered a phenomenal amount of material. We're now moving towards Unit D, which is going to be worth 15% of our diploma, and we'll start off with that Hardy Weinberg. But congratulations for getting through the largest part of Bio 30. Really heed these words. Become a master of this unit if you're looking for those super high marks. Good luck on that diploma.